I'm Barbara Engelhardt. I'm really excited to be here today. And this is an old version of the slides, so I'm going to talk you through the first two without you actually being able to see them. I apologize about that. Okay, so today I'm going to be talking about a topic that is obviously very important in approximate Bayesian inference, but it's going to be from a different perspective. So I'm very much an applied machine learning person, and what we did was we were very excited to use variational inference, types of variational inference in the models that we were looking at, and we found that they had some problems, and so then we thought about ways to fix those problems, and I wanted to share some of those ideas with you. So again, without the ideas from the first two slides, the problem that we found was that variational inference is not robust for complex hierarchical models when we fit those models to high-dimensional data. And the question that we had then was, would it be possible, how can we combine results among different variational inference estimates to make them more robust? And also, what can we say about the estimates from the aggregations across different runs? It's very specific. We're trying to solve a very specific problem. So the main goal of our work is parameter inference. We really want to look at the parameters and figure out what structure is in our data. We're not interested in prediction. So we have a very different goal. And that actually influences the questions that we're going to be asking about the methods. And the two, so one problem in this space of parameter inference then is that we see that if we find two different local optima from variational inference with the same elbow, the same evidence lower bound, they may not be equivalent in the sense that they're going to highlight two totally different signals in the data. And we'd like to figure out, again, a way to robustify those results by looking at all of the different, as many different local maxima as we possibly can. Okay, so now I can actually look at these slides. Okay, here we go. Okay, so the model that we're going to be looking at is factor analysis. And factor analysis is a linear map of high-dimensional data. So in the data that we're talking about, we have matrix Y. It's going to be P features over N samples. And this is sort of a transpose of the classical factor analysis model, and the reason is sort of for historical reasons having to do with the data. So again, the factor analysis model it projects matrix Y onto a linear subspace. In this case, that linear subspace is going to be lambda, using weights x, which we're going to call factors, and we're going to assume Gaussian noise epsilon. And the residual variance is going to be specific to each of the features, each of the P features. So when we were looking at this in the context of gene expression data, we found that we needed to include additional sparsity, and the reason is this. So if we thought about what kind of signals we're going to pull out of these data, we're going to look at gene expression levels. So this is going to be for a set of samples, N samples. We're going to look at the gene expression levels of P genes. And we found that there are going to be many subsets of those genes that are very important in the data analysis. So for example, they might be a very well-connected component in a gene regulatory network, for example. Or they may be a bunch of genes that actually covary only in a subset of samples, like only in females or only in lung tissue. And so we wanted both sparse and dense factors for genes. Conversely, we also actually had the same problem for samples. So we found that there were certain types of structure in the data, for example, variants that was only found in people who were smokers, for example, that we also wanted to subset the samples on. And so the way that we dealt with this was we put regularization on both the factors and the loadings. It ends up looking like a Bayesian biclustering model, although we also have dense factors. So the way that we did the regularization, and this isn't that important for the big message, but I just wanted to put it out there if anyone was interested, is we put a prior called a 3-parameter beta, which is a beta distribution with an additional variance term. And we actually did 3 levels of this regularization. The first level was to shrink the entire factor matrix as much as possible. The second layer was to essentially rescue a few of the factors that were actually going to be supported in the data. And the third level of the regularization is to make each of the factors sparse element-wise. And then you can see that the distribution of each of the loadings, then here, lambda, is going to be a Gaussian with 0 mean and variance 1 the variance term where the regularization will essentially shrink it to a point mass at 0 if the variable is very close to 1, and it's going to be 0 and a very large value. It's going to be a very flat Gaussian if that value is close to 0. We actually put this exact same structure on both the factors and the loadings. And that's how we get the exact same type of sparsity structure in the samples and the features. Okay, so if we actually look at how we can structure this in terms of variational inference, the regularization on X can be written as essentially 6 different gamma distributions. This is from the original paper on the 3-parameter beta process, or 3-parameter beta distribution, sorry, and where the scale parameter is the hierarchical parameter. And then what we do is we actually, in order to have dense factors, we actually have a 2-component mixture, the last row there for sigma, where we actually either draw from the factor-wise regularization, or we draw from the element-wise sparsity. And that gives us either completely sparse factors, or element-wise sparse factors, or dense factors, where all the values are non-zero. Okay, and this is actually the full 2-component mixture, where we have a beta Bernoulli as the mixture components. Okay, so when we actually fit this model to data, we can think about factor analysis in terms of a regularized covariance estimator. So if we integrate out the factors, we actually have a description of our omega here is going to be a low-dimensional estimation of the covariance matrix of our features. So this is a P by P matrix. It's describing the covariance, in this case, of 20,000 genes. And remember, this is a large number of genes, very, very small number of samples. So we do need this regularization to be able to get a good estimate of this covariance matrix. So next thing we're going to do is invert omega, and then we have the precision matrix for the set of genes, the P genes. We're going to normalize this precision matrix. It becomes partial correlation. And as we saw before, if we threshold this precision matrix, we're going to recover a Gaussian Markov random field across the genes. You can think of this now as a network, an undirected network. Okay, so we can actually take this one step further, since we have sparsity on both the samples and the samples and the features. And we can think about context-specific networks. So in this case, what we do is we actually subset the loadings so that the only loadings that are included in this estimate of the covariance matrix are the ones for which a specific context of sample is non-zero. So in other words, if we have if we include only the factors for which the factor values are non-zero, for example, in females or in lung tissue, for example, then we're going to get a covariance matrix that's specific to or only found in that subset of samples, that particular context. And when we do this, we can then recover a bunch of different types of networks, including context-specific networks, which I'm focusing on, where, again, the factor is only non-zero only for a subtype of a specific context of the sample, and then that covariance matrix is only found among genes in that particular subtype. Okay, so to do this, to do the actual fitting to the data, we use variational EM. So we can write out the variational approximation to the full distribution. And I'm just showing you what it is here. I think the highlights are basically that the factor loadings are going to be drawn from a Gaussian, and everything else has the appropriate, the easiest distributions that you can possibly imagine, including gamma distributions, the beta and Bernoulli, obviously, too. Okay. So the way that we actually run this is we perform a random initialization of the parameters just drawn from the variational approximation. And specifically, if you think about what we're doing for lambda, which becomes critical later, we draw all the elements of lambda from a normal 0, 0, 1 Gaussian. So then we iterate until convergence, the expectation step, where we compute all of the sufficient statistics. And then we perform the variational M step, which is just going to be coordinate ascent variational inference. We update our lambda variables in a greedy way. So we think about each term, each factor in lambda matrix, controlling for the ones that we've already estimated. Okay, so what we found when we applied this to our gene expression data set is the following. So we found that there was a substantial amount of difference among the runs. So what you're seeing on the x-axis is about 450 factors that we found from run number 1, we're calling it, and the y-axis is about 350 factors that we recovered from run 2. And this is just going to be the correlation of those factors, and just to make it easier to look at, we sorted them approximately by proportion of variance explained, although this was not part of the objective. So what you can see is, for those factors where there is a large amount of variance explained by that factor, they're very well correlated. So you can basically, you see a very robust estimation across the 2 different runs for about the first 100, 150 factors. But after that, where the signal that we're actually most interested in for these network recovery that we're going to do later is very different. So you'll see that there's actually very little correlation among the factors when we're looking at sort of the tail of the proportion of variance explained. And this is, of course, due to a lack of robustness in the initialization. So it entirely has to do with the fact that they were 2 different initializations. This is obviously not a good thing if we're going to use these, we're going to interpret these networks. Again, we find that we can have 2 different networks that have approximately the same evidence lower bound, and they will look entirely different. And we really need to figure out a way to allow us to recover similar substructure from different runs, different initializations. So let's do the easiest thing first. So what we tried to do, and actually it seemed to work very well, is that we would just run variational EM 1,000 times from 1,000 different starting points. And then we do exactly what I told you, which is we invert the covariance matrix and build a gene expression network, one of these Gaussian Markov random fields, from the results for every single run. So now we have 1,000 networks. And then what we're going to do is let each network vote on the network edges. So in other words, we're going to basically threshold at some number r, little r, the counts of each edge in the network. Okay? And so let me show you, or I should say first of all that these ideas about using these, combining results across different runs of particular methods is absolutely not new. Here are 3 things that I know about in the most recent literature, but obviously bagging, bootstrap aggregation, other work from some of our speakers tomorrow in the Comp Bio seminar also have very similar ideas in their work. So this is absolutely not new, and if you saw the newer version of my slides, you'd see a lot more related work. So I apologize if you were left off that list. What we do find is these tissue-specific networks. So we were looking, in this data set, we were looking for networks that only occurred in a specific tissue. So for example, the top network that we recovered is adipose-specific network. Adipose is just fat, in this case, subcutaneous fat. What you can see is, so the size of the node, these are all genes, the size of the node represents betweenus centrality. And what you can see is that even on voting, even marginally considering the counts of these edges across the different runs of the data, we find that we get this very nice, well-connected network, which is kind of surprising to us when we first saw it, but it's very satisfying. Moreover, we also found that the genes involved in this network are very related to the tissues that we were looking at. And I should say that we originally had the type of tissues switched up. So we sort of did the controlled experiment of what happens if we look for those genes in artery tissue, for example, and we did not get the same results. So this is kind of a nice thing. So we get these beautiful connected networks that actually make a lot of sense in terms of the tissues that we're looking at. A lot of them are actually involved in specific disease dysmorphisms in the tissues. So here we have 2 more lung tissue and skin-specific network. And in particular, the skin-specific network is sun-exposed skin. These are samples that come from parts of your skin that have exposure to sun. And you can see that actually most of the genes in this network, I give 2 examples here, but a lot of these genes in the network are sort of involved in pre-cancerous type of dysregulation. Okay, so we can actually go a step further. So I'm telling you that we're doing this exploratory data analysis on these very high-dimensional data. And we can actually take it a step further. Each of these edges now represents a hypothesis in some sense, and we can actually validate specific edges. So the way that we do this is we know that there are particular mutations in the genome that affect the gene expression levels of particular genes. We have them for pretty much every gene in the genome at this point, these mutations. We're going to call those cis-QTLs, expression quantitative trait loci. So if we have a cis-QTL or a mutation in the genome that we know affects gene A over there, What we can do is take this context in our data. For example, if this is a lung-specific network, we can take that cis-EQTL, that mutation, and test only for association with the neighbors of its direct target in that network. And when we do that, we can validate those edges. And we're going to do this out of sample so that we don't double dip into our data and the correlations that we know exist. And so when we do this, we actually find something that seems fairly obvious to you, but from the perspective of the genomics community is kind of extraordinary. We find many of these trans-EQTLs, or distant EQTLs, these mutations in the genome that affect gene expression levels of a gene far away from where that mutation is sitting. And we also find these in a tissue-specific way. So in other words, we have 85 of these distal genetic variants that affect gene expression levels for the fat tissue that we have, the fat tissue samples that we have. And again, if you look these up in PubMed, we find that a lot of the associations that we found are very much associated with a tissue dysregulation in the tissue that the network encodes. So that's really nice. I should say too, actually, this is the first part of the story. The biological part of the story is that we're actually solving some real problems here. So we found some of these, specifically a thyroid-specific mutation that affects gene expression levels. And it turns out that this mutation is actually very much involved in thyroid cancer. And what we're doing now is going back to thyroid cells and actually trying to figure out if we change that mutation and look in this network that we've built, how it will affect downstream all of the neighbors in the network. And we can do this in an experimental way to actually validate the findings that we have from this exploratory data analysis. And so, again, the hypotheses themselves come from this aggregated network, which still I'm kind of amazed and I don't know how it works. Okay, so we developed this Bayesian biclustering model. We're using it to identify sources of gene covariation and to recover gene co-expression networks. And we have a lot of different types. We don't know how this aggregation works. And mostly I'm interested right now in developing and formalizing the methods to be able to robustify the results. One possible way to do that is to use different types of stochasticity, including stochastic variational inference. And combining across those runs, if we could think about sort of other types of bootstrapping that we could do on these data to be able to get the additional randomness across the runs, that might be a good idea, and then formalize why they actually work. And then also thinking about methods to combine across posterior estimates with different types of marginals. So here I've described a very specific Bernoulli variable, which is with the presence or the absence of the edge. And we might want to do something more sophisticated than that. For example, understanding the entire set of factors that may or may not exist in these data. Okay, and in summary, I just want to thank everyone who's helped me out with this work, including a lot of my students and great collaborators. Thank you.
Yeah. Thank you, Barbara. And sorry that I came a few minutes late for my lunch break. Yeah, so we have definitely time for questions.
Thanks for the talk. One thing I'm wondering is whether maybe deficiencies in the variational model might, through this voting method, give rise to spurious connections, and whether you did anything to mitigate that or explore the possibility on synthetic data.
Yeah, well, we definitely looked at this on synthetic data. I actually have a bunch of results about how if we know exactly which graphical, the network that the data were generated from, we can recover that network fairly nicely. But again, if you look at the very low impact signals in that model, we have a hard time robustly recovering them. But I think your point about the changing the variational approximation is very well taken. I think we could probably do a much better job there to be able to make these models at the variational level more robust to the initial starting point. I think that's great. Yeah.
More questions?
So there's old work I think, almost 20 years ago by Tommy Acknowledgements, Mike Jordan, on basically using mixture models as your approximating distribution for variational inference. Have you thought about whether there's maybe some connection between averaging 1,000 runs and, you know, a mixture of 1,000 of your exponential families as your approximating distribution?
Yeah.
I mean, I see that what you're doing would certainly be easier, but because the mixture model was always a mess in terms of the details.
Right.
But I wonder if conceptually it maybe is trying to get at.
No, that's a really good point. Yeah, yeah. I mean, even in Dave and Matt Hoffman had a paper fairly recently on stochastic variational inference where they effectively do a similar thing. They just average across a whole bunch of random restarts as well. So, I mean, I think there's a lot of people have been going this direction for a while, trying to get these results fairly robust, and it depends. I really like to understand again why they're working and sort of how they work. I can see that the mixture model would probably be an easier way to do that because it's I think more formal. Yeah.
So we have one more question. All right, can you speak up?
So when you found the candidate, I guess, the candidate, sort of like collection meetings, is it looking you did some kind of false discovery rate to quantify that?
Yeah.
How does that correspond with the Bayesian model? Like asking the really naive person, like how do you combine those two ideas?
That's a good question. So, I mean, okay, so the FDR is basically just another two-component mixture model. This is from a Schaefer-Sturmer paper in 2005. I'm happy to share that with you. But we use that model to do these FDR thresholding. We do that locally for every run, so actually they all have a different threshold. But then if you think about it across all of the runs, we essentially have this marginal Bernoulli variable that we're also thresholding across all the runs. So I think there's, again, some layers of complexity there that we should probably model in a more formal way, but we essentially do this frequentist FDR calibration on top of this model. Yeah.
All right, thank you very much, Barbara.